Good morning, good evening. Thank you for listening. This is an agricultural, gardening and horticultural show focusing on plant science, soil science and microbiology. The information provided is for educational and research purposes and is fact-checked. Can be relied on. The websites or any of the papers discussed will be put in the description below. And today's topic is dunno yet. Dunno. So if you're already watching this back, fast forward until you see the screen change, or if it's gone for about 10 minutes, there's been no stream today or nothing really to discuss. So I'll give it the usual another 70 seconds until folk arrive. Another 55 seconds to go. Thirty seconds more. No, 20 seconds to go to try and think of something to talk about. I don't know what it is. 10 seconds. Two minutes. And I should be starting now. Giving it 2 minutes for folk to arrive. And hopefully put some stuff in the chat so we can get some discussion going or gives the show some direction. I'm looking for a topic. So if you've got anything you've heard, any weird plant science, soil science and microbiology stuff, please put it in chat. I'd love to hear what you've got to say. Because at the moment I don't have much to talk about. And today's stream isn't a planned one. I haven't got a topic for it, a preset topic. So I don't know how it's going to go. I know I can, my uni started, which has been good. Been enjoying that. A couple of good microbe classes I'm taking. Hey, Genetic Memory Farms, g'day mate. C says, good evening. Aussie CC, I just came in from harvesting cucumbers. Oh, very nice. Did you get the result that you wanted to do? Got to do a canning day tomorrow. Oh, really? My last microbiology class was on canning. That's pretty interesting. Don't get any Clostridium. GMF says A flat so far. A flat. And for all of you who don't understand what a flat is, it's a fill in the blank someone. A flat. It'd be like I reckon a 40 litre tote or a 40 litre plastic container. It's always good to do harvesting. It's so rewarding. And there's so many things you can do with it too. For instance, here you go. A flat is a square box that you would put sodas or beers on or a wrap of plastic on top. Like a pallet. Like a 4 foot by 4 foot pallet. Would that be a flat? Because if that's a lot of cucumbers you've harvested. And when you're harvesting, you're thinking, geez, I've got too much. He says, like an open top produce box. Oh yeah. So probably like ones I hold lettuce in, you picture. So something that's 1 foot by 3 foot by 1.5 foot deep. Okay. Wow. You got one of them full already. It's pretty good. And one thing that I did a course called Post-Harvest Operations of Agricultural Production. And when you get too much produce, you can always dehydrate it and make it store for longer. I always thought that was pretty cool. That's something that interests me. Don't know how I'll go with my vegetables though. I got some gigantic cauliflower, like humongous, the size of a human head. And it's an odd shape. It's got all spirals in it too. It looks pretty cool. They'll be getting harvested soon. I've got to take one. I chop one down a fortnight because I cook once a fortnight. I'm doing the canning tomorrow, eh? What slides can I show for a canning? It was just done. I haven't really collated them very well though. Don't remember which course it was. It'd be environmental microbiology. Environmental food and dairy microbiology. I'll have a squeeze. I suppose you'd like to see too, wouldn't you? I can show you. See if we get some canning. No, not that one. Share screen. I should change the view then, shouldn't I? I'm not going to see much. Better. And I don't even know if this is the right one. This is to do with food spoilage. There's a big thing on mechanical when you're harvesting. Most of this food spoilage or damage for food comes from breaking the skin. So the epidermis on the outside has been torn or broken or bruised or something like that, which has caused this process to take place. In this case, it was mechanical pressure. Because it goes into, once this mechanical pressure happens, it starts to release the enzymes, and that starts to break everything down. In this case, it's degradation by sugar, by microbial enzymes. Food spoilage. Mycelia growth and non-alcohol. No, not interested in that. Bacterial slide. Come on, canning. That's right, I got to focus. Bacterial, no. Because this is a microbiology course, I can quickly go off tangent. Gram-negative Mozilla, no. Clostridium, that's what ones are, that's from in meat, but that's what you'd expect in canning. So if you put them under a microscope, you hope you wouldn't see that. Vacuum packed, very good, come on. I'm looking for canning. Microbiome, no. Pathways. Got a bit of meat at the moment. No, we're not doing meat. It was doing with root and fruit and veg. So I should go back to that one, which is not this week's, not this lecture. All right, get out of that one. What thing I'm doing? What's this one? 20. 20, 21. Still sharing, yep. By microbes, sort of getting into it. This is a pretty rad list. Different organisms contaminating vegetables. Thanks for that. Different organisms containing, yeah, there you go. Anyway, I'm not going to go into that. I'm focusing on canning, that's right. Next. No, it's not slide showing anymore. I've got to press the wrong button. Uh-oh. Mold lesions. Talking about harvesting fruit and the possibilities that problems that can happen. I really like this. Come on. I really like this slide, that one. I like cartoons. This is just the different possible ways of contamination of leafy vegetables. Soil, manure, fertilizers, unpleasant. There's all different ways that you can get contaminated, but I just like the graphic. It was pretty cool. Talking about mechanical disruption of fruit and veg. Composition of fruit, no. 85% water, that's varying, isn't it? That's pretty in general. 30% CH, carbon, hydrogen. Oh, I'm not going into that. Some different diseases caused. Banana with no yeast, with yeast. Oh, I wonder what type of yeast. You can tell from the colours of them. See the green? That's the penicillium genus. Aspergillus, there's some black ones and some green ones. There's Aspergillus with some black. Pear south rot. Okay, yes, into with animals, Pseudomonas isolated from milk. Poor cow. Bacterial species isolated from kitchen workers around the world in different hospitals. So this is to do with the amount of bacteria species that's found in private hospitals, public hospitals, and all hospitals' kitchens. So you can see some of these are pretty bad. But this is to do with, this is around the world too, but it's in the developing world. So that's why some of these can be improved. Remember, I'm not talking about microbes. Rancid, yuck. Clostridium. Ghee. And see, isn't it amazing? Ice cream contamination. See around the rim of it, how it's changed colour? That's got a bacteria growing. Isn't that a spin out? Salmonella from ice cream. That's just amazing. Just how you package and how you take care of things, what's in the air. Contamination of fried ice cream. And the reason you think, well, how can this get blooming contaminated? It was in the oils. And a miniature crack is enough to get, and I think it was, what's the ones for the miniature crack? And I'll say it. I should remember and try and remember it first. It's no. Cladosporium. Yeah, what's the name of the species? I can't remember. Muca. The amount of colony forming units, they reckon 10 to the power of 7 is enough to get a, if you have, say a yeast, for instance, at 10 to the power of 7, you need enough in general to get sick of, which I thought was good because at least they've given a number. It's nice to have numbers to work off. Look at that, contaminant, bacteria and fungal contamination of beer. You wouldn't think it goes through the process, but yes, it's still going to bloom and happen. This is where I'm sort of quickly getting off the track here. It's supposed to be on canning. 22. Try and go fast. No, don't have it. Here we go, canned food. So Bacillus stereotherapus. Stereotheremophilus. That's the one that they found in here, as well as Clostridium's pretty bad. Desulfuryl nitrificans. So that would take out sulfur and probably nitrogen. Look at that, leaked container. That's the problem. They didn't. This one wasn't sealed. So you think, oh, I'm going to buy a can. It's good for years, and that's what I'd think too. Well, not if it's not done properly. You get these canning machines. You can do them yourself. I'm not going to go heavily into that much. Isn't that cool? Pseudomonas fluorescens, because it's actually fluorescence. Put the right the fluorescent tube under it, and it gives a fluorescence of it. And that can be used in plant science as a positive, can also be used as a negative. So it's good to know the positives and negatives about different microbes. Canning, that's it. 22, disappointed. Looks like on the milk and off the topic a bit. I'll stop and see if there's any questions actually. Oh, yeah. Genetic Memory Farm says, I will likely just do a double sour brine, so no water, add just vinegar and species and spices, heat, and jar sliced cucumbers with the warm brine. As the brine cools, it will seal the jar. Nice. So you don't put it through an autoclave, pressure cooker afterwards, do you? Because I was talking about you should pressure cook it up to 115 Celsius for like 10 to 15 minutes. I'll have to check on that. Or you can go up to the ultra high temperature one in milk, which is 140 Celsius for 2 seconds. But Genetic Memory Farm says, no, not for those. This is just a fridge pickle. Oh, okay. Yeah, I don't know anything about cooking, so I'm really sorry. I know the stuff about microbes because that's what I've taught. And just go about the possible threats that you can get from it. From post-harvest operations. Well, so it's sort of relevant today. Let's go back and have a look at, from not processing food right, what can happen if you're harvesting your food. Good techniques from many vegetables and they will keep for years, like sauerkraut, no heat necessary. Yeah, sauerkraut's using dominant microbe species to create the biofilm, and that biofilm has a lot of antibiotics in it and stops other microbes growing. That was in my industrial biotechnology class last week's stuff on that. Maybe I should show some stuff on that. I was just going to show some stuff on microbial food spoilage. What kind of microbes? Oh, you're not doing meat. Actually, you don't even want to know. You want to know more this. See how the colours you can go off? I was saying before, penicillin was green, aspergillus was black and white, Clostridium, white, black spots, white spots, Chirosporium. So just different colours represent different genuses of fungi. Cow. Yeah, I was amazed to read this about for those that eat meat. I only eat chicken, but it says the physical condition of the animal immediately before slaughter makes a big difference. If it's got disease or any infection and problems, it could be actually in the tissue, and you're going to consume the tissue. And my neighbours have extremely unhealthy animals. Well, they froth at the mouth, they cough, they got lung infections, I hear. So I thought, wow, spoilage anaerobic. The nose knows. You let your nose know and this sort of stuff. I'm not going to go into it. It was pretty interesting, though. That was meat. Now this is milk, spoilage. Check where it comes from. Then it's processing temperatures. That was quite interesting. Yes, these Pseudomonas florens can actually produce protein and lipic, so protein and lipic type of enzymes that start to break down the milk. It's not good. It's pretty. But if you're going to do if you're interested in batch culturing or doing any type of microbe reproduction in your plant science, get some milk that's unpasteurized, and you're going to get some amazing results. Most of my microbiology stuff for plant science comes out of milk, but not to say you're going to get every one out of it run every time. But it was just pretty interesting to learn that. Look at the different colours. You get some Pseudomonas syringia, yellow milk, Pseudomonas xanthia, red milk, Ceratia, marciens, and you can even get brown milk. Colour changes. Different bacterial genuses this time. Spoilage. It's pretty cool, isn't it? Might as well do. Oh, I'll do the last one then. Can't remember what it was. Bacterial and non-bacterial toxins in food. That's right. They're just inside the food or outside the food, how it gets produced. There's canning again. Homemade canned foods. Clostridium botulinum causes botulinum's disease, which is blooming horrible. It's very, very, very common. So yeah, if you don't, botulism is caused by ingestion of toxins formed by Clostridium botulinum, an anaerobic spore-forming bacilli that works inside of cans. So if you're not clean and sterilised and done all your right procedures beforehand, there's a chance spores that are alive, you got spores, and they're going to get inside here, and bang, they don't need, they're anaerobic. They will thrive. A good way to tell for that is the swelling. Oops. If you've got gas inside, most of these microbes, they off gas, and you'll see the swelling. So if you open up your cans and they go, or if they're really hard at the top, just be warned, there might be some activities happen. There's another different type of one. Causes SIDS in kids. Pretty bad, isn't it? All these types of things you can get. Salmonella. These are pathways for its mode of action. It's all serious. It's a gastroenteritis type of a thing. E. coli. We got good E. coli in our guts, you know, and there's bad strains too. Shingles, Vibrio. Okay, off the topic. Mycotoxins, the ones that come from fungus. This is pretty cool. Fluorescence aflatoxins. Aspergillus. There's an example down the bottom. See the infected apple juice? See how it's swollen? You go, oh, wow, big deal. It's just probably done something. It's got a bit of gas in it. Yeah, it might be CO2. That's because pretty much directly because of microbes in there. Anaerobic microbes have off-gassed. So it's just as bad as eating this green mold. I like these courses. They just give me a bit of an eye-opener, the stuff that they didn't teach me at school, which they should. Infected corn, fuzziness, T-toxin, fusarium. There you go. Good techniques. GMF says, sauerkraut, I need to make more. GMF says that will make your vegetarian real quick. Well, if you do, depends how you process it. If you process it right, it shouldn't. I'm happy to eat chicken, and I know how to cook the chicken, make sure it's cooked. I know the colours of it, the smells, get it from a good source. Well, a good enough source from the shops, I don't know how blooming good it is. I'm not paying top dollar for organic, but this does come under the plant science because we all eat this stuff, and a lot of people don't think about the post-harvest operations. The handling, especially at the supermarket. You watch some of the produce people, they had a big box of, what was it? Let's say potatoes. It was big. He lifted it up and he tipped it upside down and it went, and I went to the manager. I said, that's appalling. Do you know they're all bruised now? They've just started their breakdown processes. Are they trained? No. And oh, great. There's nothing, not in actual process handling. There's not much I can do. But it's a good example of, like I come home and cook things next day. So they're all right. But, and the farms that have handled these things, they would take care in all of their produce that they do as well. Just interesting facts. So we've got to eat it. We want to become better humans, and it's to do with food. And if you can get the better vitamins and minerals before they're broken down into potential sugars, there's just going to be carbohydrates and wasted and burnt off in our bodies when we could be using it for something else. It's massive. And this stuff's overlooked. It's not really taught much. Well, not that I really see. People concentrate on getting the products. Oh, look, we've got it now. It should be sweet. That's assumed. They grow in different ways. Organic grown with the different decadgens and the amount of the ORAC value ratings that are in it. There's so much higher. The secondary metabolite profile that's huge. It makes a big difference. I shouldn't go on about it. What other slides can I show? You tell me. What was those? Let's have a look at what the industrial microbiology was. This one. Week 4. Life cycle microbes. All right, let's have a look. This is to do with if you're going to, so the first bit of the stream, I'll write down, what was it? Vegetable production. So it's going to be now industrial biotechnology. All right, let's have a look. Let's have a look at some stuff that's relevant to today's topic. Hope I can find it. Sharing. That's what I was going to say. This can be done with teas. So if you're going to make some microbe teas and are interested in multiplying the numbers that you already have, this is where this course really helps. It's going to be a lot of stuff that I'll be skipping through because they're the chemostat, which goes into pretty heavy detail, this course. But there'll be some stuff that I can show you that's going to be pretty cool. If I can get to it. I might looks like I'm going to skip it for a bit here. Need to know all that. Come on, no problem. No. Thank you. This is a really flash way if you're going to make the industrial way to reproduce microbes. So if you're going to do this at home, you could add a few of these things into your system. And yeah. So the main thing is really oxygenator at the bottom and the stirrer. But it's more so the bubbles. See how they're distributed? It's called the sparger. They're to do with also the size of the holes, to do with the size of the smaller the better, because the big bubbles, believe it or not, they actually can kill microbes. They burst and stuff. Yeah. So that's really about it, and maintain sterility. That's all I probably want to mention, because I'm not going to go into much detail. I just want to show you some cool things you can do to help. Stuff that I picked up, because they show the stuff that they do, which is pretty cool. That little green one that was in the IoT lab. Okay. Different stuff you need on them, and why you different stuff. There's the sparger, used for aeration. For the purpose of the aeration, to provide sufficient oxygen to the microbes for metallic requirements. Spargers can be of three types: porous, orifice, or a nozzle. The porous types are those, the ones that you see in fish tanks, for instance, where they put out the micronized bubbles. Which is pretty cool. Just got to clean them. This is going to be important. I think pretty much that might be about it. To save, if you've got any questions, whack the questions in. Yep. It's all to do with ethanol fermentation. Okay, we're going to get into that, are we? This is at the industrial level. So if you can scale this down, you're a winner, because they've done a lot of, many thousands of hours in research to find out the best ways to multiply microbes. It's business. There's some links that are cool. That'll do, I think. Into the air. I like the air bit. Airborne microbes. What size filter should you use? Well, it's going to be under 0.5 micron. Modes of sterilization. This is pretty cool. Filtration, UV light and heat. Filtering, filters. That'll do. Stop sharing. Very good. Very good. TMF says eating better and more nutritious means being better caretakers of soil and water. Yes. Love your pond, by the way. Thanks, mate. Yeah, put a lot of work into it. That's right. We've got to maintain everything, not just us. Well said, mate. That's right. That's, yeah, by eating better, my pets eat better. I've seen spring has sprung here, and I've given them a lot of fruit and vegetables, and they're running around like anything. Not fruit, a lot of veggies. My goats are really happy. I've seen the difference. So they must have got a few little things they are lacking, and it's really good to see. It's not just me too, my pets. And then the land will prosper, maintaining that, keeping it right ways. It's cool as. There's lots of cricket on this week. I know people are interested. Woohoo! Australian cricket starts in 10 weeks' time after my uni finishes. I'm in the 5th week of uni already. It's bloody excellent. I'm enjoying it. One of those classes too is called the Science of Happiness. Can you believe? The Science of Happiness. How cool. Just started that this week. It's an 8-week class. I'll probably sit the exam for that one. When we go back to its plant science or science and microbiology. Yay for microbes. And the pond. Yes, even in the pond I'll put beneficial microbes too. I'll put ones that you put in an aquarium to take out the nitrogen. I can't remember the names of them. Yeah. Even trying to take care of them too. Yes. Yes. Goat fun. It's fantastic. Spring sprung around here, so all things are starting to look for nests. And it's good. So the plants, the seasons are coming in. That means the time to get ready for seedlings is even happening. Poor people in the northern hemisphere, it's the opposite. It's harvest time for yous. That's right. That's where we were going into, harvest. And dehydration, that's where I was going into, that mode. Got fair dinkum distracted. But yeah, the post-harvest operations, and I was going to say about if you get too much excess produce, you can dehydrate it and save it for better times. Or you can do what GMF's doing today and canning. Cool. Something I've never gotten into any post. I've never had an excess of produce to do anything with. Probably this is the first year, and I've given a little bit more to the goats. I've got excess cauliflower. I'll be eating that out of my garden until Christmas time. Right out of season. And post-harvest operations. If you get too much, yes, share it with your neighbours. Store it well, and don't bruise the fruit. The colder temperatures. Should I go through some more slides on post-harvest operations? Would you like to see them? Because by storing it, it reduces the activation time of all of the different enzymes and microbes that are inside of it, so processes will slow down. Oh, there's Josh Stokes. How you going, Josh Stokes? Nice to see you, mate. Do you have any research on living soils and biochar? Yeah, heaps. I only support living soils, mate. So all of my techniques over the past many decades have been living soils, and that's what I talk about. Plant science, soil science, microbiology. They are pretty much all to do with living soils, because living soils, microbes, and soil science is living soil, because you've got to have all the organic matter and the constituents and the right cation exchange and the redox and all that sort of stuff. And with the plant science, that comes into it, how the plants uptake it and getting the right roots and the right conditions of the soil to make that happen. So I've got stacks on living soils, mate. And biochar, I've done many episodes on biochar. It's all pyrolysis. It's all about putting the oxygen back into the organic matter to have little homes for microbes. It's pretty cool. Oh, sure, says GMF. Josh Stokes says, heart. Very good. Yeah, that's all that I'm about, mate. I'm not into organic. I'm not into non-organics or synthetics or any type of poisoning. I'm into mineralized from immobilized matter, not just giving mineralized as a supplement or a main fertilizer like most people do. It's very hard to grab your head around it, but once you get your head around it, it flows and it works within itself. It fixes itself. And then the microbes come into it, and then they take care of the things. I use specialized microbes because I've been doing this for years now, and I source them and get different strains and use them to do different tasks for me. I enjoy it, and the results speak for themselves. If you ever look at some of my, I've done a couple of world firsts even. It's one of the latest is producing the plant hybridization, producing Brussels sprout and broccoli out of one seed. So you'll plant the seed, and out of it will grow one stem, and the head, top of it, will have a broccoli on it, and then the side shoots will have Brussels sprouts on it. It's fun because we can all do these experiments ourselves, and it's don't have to have much money at all to do it. I'm not very rich. I struggle. So it's very rewarding for me to get extremely good results from my education. And that's why I do this stream, because everything's working. I still study at university for these 3 topics, and it's just blooming excellent, mate. So I'd like to help out others that struggle but would like to get the results that I get. That's why I do this stream here. So yeah, if you've got any friends or foe like that to struggle and got all these myths about plant science, soil science, microbiology, here's the spot to ask the questions, and I can show the slides from my university and shed some light on the topics. So there you go. Josh Shake says, thanks heaps. We'll go through and watch all your videos. You don't have to, it's up to you, mate. But also put instead of watching all of them, well, you can if you want, thanks, but they're put into a search engine. For instance, seeds, and it might come up that, or actually, what's your background, Josh? What are you into? Because there's some other stuff that other agricultural plant science things that I do on another channel that you might be interested, but that's not for everyone. So I'm not sharing that with everybody. But yeah, plenty of, save your questions, Josh, for another stream. I do this stream once a week, and I'm looking for actual titles for the stream. So for instance, next week you can come on and go talk about let's talk living soils for this stream, and just ask me lots of questions on that, and I'll show the slides. Or biochar, but biochar I don't know, it's pretty summed up. It's how you make it and what you use it for, I can nearly tell you right now. It's not much. But to elaborate into it, I can really elaborate because I got the slides from the universities to do so. And it's not from my knowledge, I'm just adding to the slides so you can see for yourself what they say, because I don't spin. It's not bro science, it's grow science here. Hello, Frank Cannon Lounge. I will never get off the blue juice Miracle Grow. #1, LOL. Yeah, Miracle Juice, when you put it in the water, it goes blue. And there's nothing wrong with that. There's many different ways for people to grow, and I'm not about to tell you which way you should do it. I support organics and non-pesticides and non-spraying. That's all. But you can grow whichever way you want if you're getting results, because there's so many different environments and reasons why people grows their own way. And that's awesome for them. I'm happy for them to do that. So good on you, Frank. Keep up your good work. With that, though, you can stop Miracle Grow. It holds a lot of the salts. It's very high in salts. So you can hold back a bit in the last few weeks of your fruit. That's for sure. And you'll still do fine. But yeah, everybody for trying. That's why here I'm not about to tell anybody, change this, go do that. No way. If you're growing, keep up the good work, and you'll learn from your experience from your growing. Oh, I might try this next. Oh, I might try with more microbes. Oh, I might see what this living soil business is all about, and slowly transition. Because you can recycle. People might be living in flats or units. They can't have a compost heap and recycle and that sort of stuff. So there's all reasons why people do what they grow the way that they do. Support every single different technique. Everyone grows and they learn from it. And then it's not a failure, it's a learning exchange. It's blooming excellent. Frank's Lounge says, LOL, I have nothing against organics. I just love trolling people with the facts that I grow, Miracle Grow, most people hate it. Yeah. See, most people have that's their opinion. I support that. Good on you, mate. People are going, listen how weird he is. You're expecting me to bite back? No way. I'm not biting back. I'm not putting someone down for growing. I think that's excellent. Everyone's got different budgets, different living conditions, different abilities. One person I know is handicapped. He gets his partner to do most of the growing, so he's appreciative just to actually get some sort of fruit. So it's pretty cool. Frank says, yeah, just wanted to fight. Yeah, that's because a lot of people, they're you have to grow their way, mate, to succeed. But I study at university and I study science, plant science, soil science, microbiology. So there's plenty of ways to do techniques. There's techniques you can do that are very expensive, that are very cheap, that blah, blah, blah. So, yeah, you've got someone here with a bit of knowledge, mate. So you might need to go to a stream where people are a bit more biased and you have to grow their way to get their results with their formula. Sorry, Frank. Can we at least pretend to fight? That's funny. Good on you, Frank. He says, oh, I know you. I've been watching you for years. Oh, okay. You're the super science nerd on plants. Oh, good on you, mate. Positive feedback's always welcome. That's good. It's because of this study that I enjoy, and I've had a lot of experience in the past, and I've gone and got experience now through university, and the combination's just outstanding. The results I'm getting is phenomenal. I've made a business out. Now I'm a garlic farmer. I've going into the third season, next season of organic garlic farming. So I'd like to help others succeed and do the similar sort of route, mate. Something legal where they can succeed and have no stress and worries, and know that they're helping out the environment and the earth, like organics. Or, for instance, you can do it your way with your blue juice. There's plenty of ways to grow and plenty of budgets and reasons why people do things, but I support the organics way. I thought about that. Is that Duckfoot, Frank? I did. It is you. Wow. I hope you're doing really well, mate. You're one of the better fellas. Do I remember? Into his genetics, and actually knew some good stuff about genetics too. Was interested in mutant breeding and getting different expressions from mutants, and that's outstanding for people to try with different types of new parameters. It's so yeah, you were the 1%er, the person you're a science. You'd probably love a lot of these courses I'm doing, mate. And most of them, you can do them for free. I pay $50 Australian for the exam. You'd pay probably $30 US for the exam, and it's done online as well. You're probably one of the ones that were interested in this. Frank says, yeah, you're doing good, thanks. I hope everything's been going good for you too. No, it's average, mate, I can do and say. Actually, you'll like this here, Frank. Now I know it's you. I'll get my what's it called? Off. I'll get you this link, off-grid. I just don't know what I'm blooming my name is. What's it called? Oh, yeah. That's not it. Had this a few weeks ago. I had a problem with that. There it is there. Is that it there? Yeah, that's it there. Okay, here's something for you, Frank. I'll delete it. But get that. That's my other channel. And that's, I post daily. And it's a similar sort of thing. It's to do with homesteading, though. But it's all to do with plant science, soil science, microbiology. But there's a bit of other stuff to do with it. Okay, cool. Good on you. Thanks, mate. This channel here got a bit stuffed up with people trolls and all sorts last year. So, yeah, I started another channel. Frank says, I have some beautiful plants doubling recessive freak show with a double recessive Doug foot. Holy mackerel. Freak show is another mutant. That's cool. You got it all happening. That's the way. I've gone off from that side of things on this stream now, and I've focused more on the plant science, soil science, microbiology. See all the pictures there? I've got better monitorization. I'm not getting strikes and I'm not getting any problems. I still talk about those types of topics, but I just watch what I say. But I'm also focused on these three now. And last week I gave a bit of a demonstration in my garden, veggie garden, on disease and microbes and stuff like that, what to look for and telltale signs. So because I struggle these days and what to talk about, mate. And I stick with getting bans and strikes and things for it. So I don't really like that sort of stuff. So yeah, we talk about those sort of things, but just in different words. But Josh says, have you seen any of mutant genetics? No, mate. No, I haven't. I don't really watch that sort of stuff anymore, to be honest. Sorry. I'm a full-time uni student, and I've got on season 3 of a garlic farm, and I've got a little bit of soil remuneration to do as well. So my time's pretty much taken up. And I'm a massive cricket fan, huge at cricket, love the 2020, and it's all starting up again too. So, sorry, mate, I haven't seen any of his mutant genetics. Actually, that might be, Josh, a question for Frank, because I'm sure he... down the bottom. Oh. See, that's why I don't read some stuff too. Good on you, Frank. Oh, gee whiz. Genetic Memory Farm says, looking at how to cheaply and efficiently build a Wallapini in-ground 4-season greenhouse. Oh, it's an in-ground 4-season greenhouse. Holy mackerel. Is that a subject for your course cover? And would you be interested in doing a series on plant life cycle? Yeah, plant life cycle. That would be all right to put it in layman's terms. It's pretty straightforward. Even my plant development biology class, that was a 4-week course. So, and that covers pretty much the rhizosphere. You go into the roots, and then you go into the phyllosphere above the roots. Yeah. But I don't mind doing that sort of thing, mate. It's always good. And the Wallapini sounds cool too, because I studied atmospheric science before I did plant science. I studied atmospheric science for probably 10 years. I've done it through graduate school in, where's that? Was it in Colorado? Anyway, I've done it a few different places. I've done it once, got a certificate in England, did it at university over there. So I've done that for a while too. So I do know a fair bit about the weather. And a 4-season greenhouse, mate, that's rad. You're talking about money though, because to create different environments, you've got to produce that. And if outside season is in winter and you want to produce summer, aircon or heater and vice versa, you've got to do that. So it's hard. I did set up a greenhouse in Canada, professional one, and took quite a few $1,000s to do it. Worked unbelievably in the end, though. Actually whinged about the electricity bill, but doesn't matter, you can still do these things. Frank says, if there were any good, I'd be working with them, and most of them are trash. No good. How to, from seed to harvest, or processing from seed to harvest? I really don't know, mate. That would be nearly one I'd like to do with you. Have a 4-part series with you, or even a 2-part series where you can sit and ask me questions and you can prompt me, because I'm not sure how to do it. I just really start from right back at the seed and go through probably to, I suppose, to harvest, if that's what you'd like. Yeah. It'd be all right. Frank says, oh, yeah. GMF, degree, tomatoes, cucumbers, squash, etc. On the Wallapini, it uses passive heat. Oh, yeah, because you've got active heat, which uses from electricity, and passive heat uses from like solar radiation from the sun's rays, or you might use it from storage somehow. So you might have water that's heated through the day, and it might be stored in the walls through thin cavities that you might set up a big water container. So that's the passive heat that you've got, which are good options for cold environments. My friend had one in the Arctic when I spent 2 winters up there. He had a really rad one too, actually. Report, just moderator, there we go. So yeah, that would be pretty cool. Also to do the greenhouse setup. Probably need more questions on that, but that'd be right, that'd be my forte. I'm a licensed electrician as well, so I can give advice on different power consumptions on different devices to get there. But passive heat is used, well, temperature comes from 3 ways: convection, conduction, and radiation. So we'd be either using 2 of those ways, because you wouldn't be using the convection, you wouldn't be heating it through a heating source. You'd be using solar radiation, and then conduction, which it touches on the walls. So you might do something like that. So I really like that. That's something that if I had money, I'd like to set up too, mate, like for real. A legit greenhouse that's environmentally carbon neutral. So you might have a solar panel sitting next to it, which might run some lights inside, or they might run some water timers or some fans. So it's, yeah, the carbon neutral thing is a must these days, and that's what this show is all about: organics and carbon neutral, and respecting the environment. It's very cool. It's the more I study it, the more I see how easily we can achieve it. It's marketed wrong, and some of the products are expensive as. But I've got an off-grid garlic farm. It's off-grid. I irrigate in summer times off-grid. It's it can be achieved. So that's the education that I support and promote, where everybody can grow all different ways for different reasons, but that's the one I chose, and that's the one I really like. Frank says, I would love a solar-powered greenhouse. Yeah, easily done. Yep. I'm a licensed electrician, so I can get all the good prices and stuff, but it's way cheaper than you think, like a lot cheaper than what you think. And solar panels are good for 25 years. So they sit there for 25 years, no service. Where the wind turbines, you got to service them, their blades, every 10 or so many years, they run out. You got to replace them. There's Raptor. G'day, Raptor. Yeah, carbon. Yeah, you're into that, aren't you, Raptor? You're a carbon neutral. Did you get your azadirachtin planted? as a director. You know what I mean. But for everybody, did you get your neem tree in, mate? Frank says, what do you think about fiber optic LEDs? Holy mackerel. That sounds awesome. That's what I think. Don't think I've heard of it. But LEDs are cheap, and then you go down the route of putting light through glass, which is fiber optic, and it's cheaper again. And it probably would, that's how the internet runs, on fiber optic. So that's ingenious idea, whoever's done that. Their heat load would be way lower, and the electricity usage lower again than LEDs. That's the next level. Wow, nice one, Frank. Oh, from the sun. So run through. He's commented down the bottom. I've seen a few companies running solar-powered fiber optics LEDs. They're stupid, ridiculous, expensive, but it looks a genius. By the sound of that, all they're doing is amplifying the sun's rays through a tube. So that's old technology, to be honest. They've done that with. Have I installed any of them? What do they call them? Sky tubes. So that's just a skinny sky tube, from what I think, from what I hear. Mr. Raptor said the panels being tested have insane efficiency. Oh, those LED panels. Okay. Yeah, I don't know anything about it, mate. It's cool to hear new stuff. It sounds really good, though. Had to put it on hold. Caretaker from a sick mother. Oh, is that why you've been out, Raptor? Sorry, mate. Hope she's doing a lot better now. She might appreciate some of this food stuff I've been talking about, my food science. I'm taking food microbiology class this semester and industrial microbiology class, and that's talking a lot about to do with sicknesses and things. So I hope she can get better, and you keep up your good work, mate. Frank says it's got a big dome that captures the light. Yeah, it sounds like those sky tubes. The sky tubes are just make your own. Put a glass tube on the top of the roof, get a flexible duct that you have for your indoor vents, air conditionings and stuff, and that bounces the light around so you can bend them. So if you don't put it right above where you want it, you can take it left or right and reposition it, and they mount in the ceilings. So they're pretty cool. Just make your own. All you'd be doing to make your own is buy ducting. So buy 8-inch duct by 20 feet and cut it up and install it. Try and have as little bends as possible, radiating the heat down. GMF says, sorry to hear that. Wrapped up. Hope she feels better soon. Yes. There's no good about that, mate. It's just putrid. She's like, oh, that's the way. It's a positive. Hey, speaking of positives, one of my courses this semester is, I've been wanting to do it for years. It's called the Science of Happiness. How cool is that? Frank says, yeah, but this is fiber optic. It runs through cords. Well, you get it and act upon like you just get the cords yourself and make them accept light. So set the cords up to like in a big dish where it might be a dome, so all the light's bounced around, and it can be bounced around into the cords. You can do it, Frank. You're very handy. I really reckon you could do it. All right, fiber optic cord. No. Yeah, because I've installed fibre optic cable before, little green stuff. It's a pain in the ass to work with. It's got a bending ratio. If you bend it too much, it breaks, it shatters because it's made of glass. So it's not real fun to work with, but yeah, I do. Frank says, I do believe I could make one, just it wouldn't be cheap. Oh, yes. All right, let's have a look. Let's get it cheap for Frank. So you go and get a share screen. So you get a 8-inch. You get one of them, 8-inch air duct, whatever size you want. Then you get some something like that. Fibre optics that's in a conduit itself, and then you'd see how it would work. But to be honest, I wouldn't worry about that little tiny stuff. They've gone and fine-tuned it. I just get the duct, mate. Eight-inch air duct, skylight. Now watch skylight. Look at this one. That's insane. There you go. That's what I was thinking of. This thing here, exactly that. See, stick it there. It's got the tube and it shines the light. You can move it wherever you want. Their difference is that they've fine-tuned it and they've got it in little glass tubes, spitting it out. And it might change the colours because they've got different glass fibres inside of it. But this is pretty easily. You just get the duct itself, mate, and a bit of glass on the outside or a dome if you want. Caravan hutch. There you go. And I'll get you a little bit of extra light to your greenhouses or to your areas that haven't got natural sunlight, or they get too much sunlight and want to move it. It's a cool idea. And all you need to do is just get that and water seal it on the roof well. Roof and gutter silicon is what we use in the trade when we work on roofs. It doesn't fail. It'll last about 20 years in Australian sun. He says, yeah, it's not the same. Oh, well, I tried. They might amplify the light, and they've done it professionally, but I was just giving you a suggestion, just something that you might be able to make yourself. It sounds like it's next level lighting. They've gone from metal halide to LED to fibre optic now. I suppose I'll put it in there. Fibre. Oh, it'll give me those little tiny things. Yeah, these things. My problem would be about the output at the end not being strong enough. All these are pretty dim, and from my experience too, they're pretty dim. Not to say they haven't changed the technology, but I don't know. I got no experience with them, mate. First time I heard of it's right now. That's what I love about this show too. You folk teach me. It's blooming great. I try and help out with my plant science, soil science, and microbiology. And you shed new light on different things for me. It's very cool. Frank says, you look for solar fiber optic, you'll come up with an optical lens that magnifies, which then hooks up to the wires. Ah, there you go, magnifies it. Magnifying lens, that's pretty clever. New ways just to refract and get different ways you can utilize the light beam. But you can't ever increase it though. There's only so much energy you can produce. You can't go and, unless you amplify, it's the word I was looking for. It's hard to amplify it. Need the word solar. The testing that they have performed with them actually shows a great growth rate and performance. Oh! He said I put solar. Here we go, look at these powerhouses. I found it, Frank. Solar lighting system. Convex lens that focuses motor, optical fiber. Oh, wow. Bloody excellent. Love technology. See, science is just produces the results all the time. I blooming love it. This is why, what was that fella was speaking before? He wanted to debate. Josh. That's what Josh says. There's so many ways to grow, mate. And they're always coming up with new and improved ways just like this. How rad is that? So you'll have these generators on the outside of the house, and inside they'll just be producing light for free. That's the future. Or that's the next step in our future. Do you know that I'm getting right into, I don't know if you're going to like ancient aliens, and it's fascinating, our future, that aliens have had technology for millions of years, and we've had technology for a few hundred, and just the leaps and bounds that we go every 5 or 10 years, I couldn't imagine it going over tens of thousands, let alone hundreds of thousands of years. So all this stuff just blows my mind. It's so cool. I love this. The testing that they have performed actually shows I said that one. Lemon Road. All right. Well, I hope next week you can come in with your cool suggestions. That's about it for today's show. Been going for an hour 13. Thanks, everybody, for listening. It was really good. Hope you enjoyed it like I did. Save your questions. And GMF had some good suggestions for some streams coming up soon, so we might be taking up some of those. And it was nice to see you again, Frank. Drop in same time each week, mate. I've left it at the same time because there's some really cool people that drop in regularly. So it's nice to catch up with them too, like yourself, like everyone else today. GMF wrapped up. It was really, really cool. And there was Josh and a few folk that are listening that don't usually comment there in the background. G'day to them too. Hope you enjoyed the stream. If you've got any suggestions for the shows coming up, just whack them in my YouTube or let me know next week. Frank says later. I'm going to say later too. Enjoy the cricket. Woohoo! Go the cricket. 2020 matches. Yay. Happy growing. Happy breeding. Good health to you all. Thanks for watching. Press like if you like. Press thumbs up. See ya.